Dr. Elias Nyanza. Hi, Elias. Great to have you with us. Elias is a population and public health specialist working on planetary health in Tanzania. Through research and community interventions, he focuses on the reduction of harmful environmental exposures that affect marginalized populations. So Elias is holding several hats, several functions, but one of many is that he's a SOPHEA project coordinator in Tanzania. So Elias, great to have you with us today in this session. You're welcome to share your slides and start the presentation.
Thank you so much, Sabine, and the rest of the team, and welcome everyone to today's session. And my presentation is focusing on unveiling urban contaminants. And actually here we are looking on ecological approaches that can be used in the remediation of antimicrobial in urban, but also in rural receiving waters and wastewaters. So we are presenting an experience from Tanzania. It's important to note that whichever that we consume or release to the environment, at the end of the day, it comes back to ourselves. So to tend to familiarize with the antimicrobial that we are talking here, this actually will refer to substances that inhibit the growth of microorganisms or kill them outright. And it can be used in agriculture, in the human medicine, but at the end of the day, we have a lot of waste that goes into the environment. And at the end, actually, it comes back in terms of bioaccumulation and biomagnification. So it includes antivirals, antibacterials, but also antibiotics, antifungals, among others. So they are all actually, if they do not respond to a specific treatment that you are doing, then we end up calling them as a antimicrobial resistance. And this now is becoming actually a global challenge. And if we do not protect our environment, that means our environment in the future, we are harming it, but it will be harming us in one way or another. So if you look on this particular diagram, it's complex, but it's showing the entire paradigm in terms of whatever we use in the treatment, in the pharmaceutical industry, in the agricultural industry, in the environmental protection. And if we do not dispose it properly, it goes back into human being, it goes back into animals. And at the end of the day, if whichever that we take as food, whichever we take as resources to eat and so forth, it comes back, affect the ecology, but in the near future affects ourselves. So you can see that the global production of antibiotics actually is around 200 tons, and actually most of it is released into the environment, and at the end of the day, actually we end up consuming it and affecting our health well-being. So you can see, for instance, in Africa, if we talk about the antimicrobial resistance, so it's happening in most of the countries significantly. So affecting animals, affecting plants, but also if we look in terms of sediments, so we have done a number of studies in the country, but also in different parts of Africa, showing how much the contamination is to animals, to plants, to soil. That means whichever that we are growing into our environment is actually taking this antimicrobial residues, and it comes into human being as food. So you can see here, if you consume fish, for instance, if you look, for instance, in Tanzania, in Kenya, in Sudan, so a lot of contamination is happening there. So if we look again in terms of meat and milk, so you can see in South Africa, in Djibouti, in Egypt, the number of food that is consumed actually is contaminated with antibiotics. So the antimicrobial resistance now, we understand is a natural process happening, but the way how it's happening now at a speed that we cannot control. And there is a need actually to address in a different way now. Instead of only doing health promotion in terms of minimizing misuse, overuse, over-counter medication, but we need to ecologically, innovatively come up with interventions that actually will slow down the generation of the antimicrobial resistance, especially in low and middle income countries. So you can see in clinical setting, for instance, there is a lot of misuse, and they already overwhelmed the health facilities in Africa, and then encompassing with increased non-response to medication associated with antimicrobial resistance is ending up affecting a larger population in terms of the disability adjusted to life years. So a number of people are hospitalized, but there is no cure because the medication that's supposed to treat is not working, but what is working now is more expensive than how one would afford. So you can see, for instance, in Tanzania, in 2019, only more than 12,000 people actually died because of the antimicrobial resistance. That means they were at the hospital on time, but the hospital could not provide the medication for that specific disease. So what are we suggesting now? We have come with what we call a biopolar adsorption. These are ecological, they are plant-based, and the biopolar material are biomass-based material delivered from renewable resources, and you can get it from plants and treat them and use it at the waste stabilization ponds. So before you release the effluent coming from hospitals, from pharmaceutical industry, from home and whatever, that is treated through this ecologically friendly material and absorb the antibiotic residues. So once you absorb, that means whichever that is going into the environment is minimal in terms of hazardous. So if we look, for instance, in terms of the testing, so we wanted first to know what do we have in the environment. So we found actually that most of the antibiotics that are dispensed at the facilities, at the health facilities, are actually into the environment. So we found, okay, fine, let us start with one after another. So using evidence from elsewhere, from other countries, so we were able to do the examination of the wastewater, of the soil, and some of the plants actually, to see what is existing within the environment itself. So this is the one of the biopolar material. This is the jamum seeds. So jamum seeds are edible, so you can harvest it, extract the juice. That's one of the economical benefit. But then that means in planetary health, you encourage more planting of the jamum plants. So that means you are conserving the environment by planting more trees, which are beneficial to the population because they get food out of that, the juice. But the seeds now here is what we are interested in here. So this seed here is dried and pulverized to get the powder. Then the powder here is calcinated at a temperature of 400. And once this one is calcinated now, this now becomes what we call the biopolar material. So we're talking about the geranium seed as the biopolar material, which actually ends up absorbing at different pH and different temperature. So what you are supposed to add now here, that different antibiotics are absorbed at a different pH. And that was a big challenge. So you have to have a series of waste sterilization ponds immediately after the filtration. So in this specific case, for instance, some of the antibiotics are absorbed very well at a pH of 0, but some actually at a pH of 13. So that means you have a first cereal pond at 0 pH, and then the next one at 13 pH. That means you can absorb most of the antibiotics residues that are coming from the waste or from the water that is received from the health facility, pharmaceutical industry, from home residency, among others. So you can see, for instance, we customize it for flaxseed, but also from the lamivudine. So at the end of the day, what we want actually here is protect the human being. That means they have water for the for irrigation, which is safe from antibiotic residues. We have water that is used for human consumption, that is free from antibiotic residues. In that way, actually, you are having healthier environment, but also healthier people that are consuming something which is not toxic, or that's something that will not harm them in the future. So I have already mentioned about the prevalence of different antibiotics residues in the environment, but you also went further to look on what kind of microbes that are available but cannot be treated, that are developing resistance in this particular environment. So you can see from here a number of them were documented, and it's not my intention to overemphasize on that. So you can see here different antibiotics were applied to different particular microbes with tetracycline, gentamicin, and you can see what is happening there. We have a lot of resistance that's happening in this particular environment, and we need actually to address. And the use of biopolar material, ecologically friendly, but also available locally at low cost is really working in Tanzania. So we have started with this particular work in a small scale, but in our near future, depending on grant availability, we'll be able to scale up now to different water stabilization ponds at different regions, so that at the end of the day, we protect the Lake Victoria, where actually SOPHEA is working. So these are just some of the examples that we use for ciprofloxacin, penicillin, tetracycline, where actually we are trying to see how efficiently the biopolar are working on different antibiotics that's available. The good thing with the proposed jamim seed biopolar, it can absorb the antibiotic residues even at trace amount. That was really encouraging, because we had thought previously maybe it's only at high dose, but even at low dose, it can be able to remove from the environment. So you can see the functional groups at different temperatures behaving, okay, from the customization of 400, where actually we develop the biopolar material, and you can see how efficiently in terms of the wave number and transmittancy of the antibiotics that's absorbed at different temperatures. So the surface morphology, once according to Darwin et al., once you calcinate it at 400 temperature, and here if you do it taking a significant look at it at a microstructure, so you can see these are the structure inside the bipolars, then you can see how they can absorb the antibiotics. This is the nature of the jamim seeds and how they can absorb the different antibiotic residues in place. So that's from the microscopic view of it. So you can see how the peak on the machine, if we want to now analyze it from the biopolar, what has been absorbed, this is how using the CMD EDX image, and you can see at a temperature of 600. So, for instance, if you look at this particular antibiotic, removal efficiency observed, so the 5 experimental runs that we did at first was at 31 to 84, but we were able to optimize it up to 94%. So you can see, for instance, for Cipro, that is slowly, but optimizing at different pH and different temperature. So you play on that until you have the optimal results. So now actually we are going even beyond 95 in terms of efficiency. So now in the field, we hope that it will be maintaining at least 95 to 96% efficiency of this biopolar material. So the best results obtained at 8 pH, but with low initial concentration was less than 100 parts per million. So this particular model that we are using, as I mentioned earlier, it's actually advancing, and we see that it will be able to maintain above 95%, close out throughout our experiment and the field. So you can see from the statistics, the F value, implying that the model was significantly, and it can be used to explain how this jamim seeds biopolar is working. And you can see the R squared 0.99, indicating that this data fit well in selected model and among others. So you can see here different parameters that need to be considered, but most important is the temperature and pH, but also the time frame that the water will be passing through the residue. Normally, in this particular experiment, we are using 30 minutes to 38 minutes to pass through the biopolar material, and that's the efficiency of 94%. So this is the calcination, and you can see how drop for Cipro, for instance, is antibiotics, how it's dropping very well at a temperature of 400. So you can see once you start at a temperature 100, slowly, but once the biopolar are at the efficiency, and that's the 400 degree, you can see the Cipro removal is very high. That's why the calcination temperature is very, very important. Once you calcinate it at low temperature, the absorption will be minimal. Okay, so these two parameters are very, very important, especially for Cipro and the APBR trademark. So remember, is increasing the surface area that matters a lot. So presence of antibiotics residues in water provides an opportunity for us to work on this efficient methodology to be able to remove. And then the geranium seed biocladrobin has the potential properties, and they can be used in the removal of ciprofloxacin and lamivudine, among other antibiotics. So for now, we are confident with these two, but we will keep increasing in terms of other ones, so that at least we are able to remove most of the antibiotic residues at the different environmental conditions that are needed. So for now, we have optimized for these two, and now we are keeping now on looking on how we can optimize for the other three. And maybe in the near future, before we scale up, we will have already have the optimization for most of the antibiotic residues. So these are the papers you can read at your own time. So we did a review, we did a contribution of these drugs, but we also went to look on what is existing in the environment. But we published also on the removal of lamivudine from synthetic solution using the geranium seeds. We published this specific data as well, but in the way how we can optimize. So you can read at your own to be more consistency with what I have mentioned in a nutshell. Thank you so much.